It's 3 o'clock on the dot. So as people filter in, we will, I think, just get started, and they'll join us when they start. So wherever we're at. So I'll just turn the floor over to you guys. I guess the only logistical thing is that we will be, I'll just ask that you guys send me your PowerPoints basically like 2 weeks in advance. So let's say 5 p.m. on Thursday, 2 weeks before you give your presentation. Next week, we'll do a presentation with Dr. Wu on Gamma Knife. So that gives everybody a little bit of time after you see, I work with Andy and Calvin on this presentation. So you can kind of see the way that we want to do it. You'll basically see that it's basically a book report of the chapter. It really like uses the same terminology and outlines basically exactly as the chapter does and follows in a very linear way, exactly like the chapter. All the different things. And the great thing that they did is they integrated the additional details that are in the figure legends, and they included the figures, even if they appear on different pages, but they included them with the relevant parts of the text when they're discussed. So you'll see kind of how they go through it, but I thought they did a really good job putting their presentations together. So I'll turn the floor over to Andy, and if there's any questions, obviously we're here. Emily and I are here, and we'll continue to filter in, okay?
Great, thanks. So I'm Andy, and I'm working on this presentation together with Calvin. I'll do the first half, and Calvin do the second one. I'm a second year at Tufts, and here it goes. So we're starting off with the clinical vignette. Feel free to add any input as we go, by the way, or pose any questions. I'll also be asking some questions, so just feel free to unmute yourself to answer them too. So first of all, we have a 79-year-old male who becomes forgetful, is misplacing items at home, becomes confused when paying for groceries, and has generally worsening cognition. He's oriented to person, place, and time, has normal speech, is unable to recall 3 unrelated words 5 minutes after repeating them, and is slow and difficult with adding and subtracting numbers. Can anyone tell me broadly what they're trying to get at here? What diagnosis? See if we can.
Alzheimer's.
Yeah, Alzheimer's. So one of the most common causes of dementia in the elderly. And yeah, just feel free to chime in. Here we have a side-by-side photograph, and you can see that with the Alzheimer's brain, the cortex is has widespread atrophy. There's a narrowing of the gyri and widening of the sulci. So here are some MRIs in the transverse plane, and just since this is a very basic presentation, we'll just start from the very basics too. This is T1 weighted, which means that the white areas are includes fatty substances, skin and bone. The black is CSF, and the shades of gray are the gray and white matters, generally speaking. So this is a slice through the anterior horn and atrium of the lateral ventricles. And you can see in particular that there's a dark region as a result of the thinned cortex and greater CSF compared to the normal. And this is really characteristic of Alzheimer's. You can see that the ventricles are dilated in the sick brain as well compared to the healthy. Moving inferiorly to the brainstem, you can actually see that the brainstem is not as grossly affected as the rest of the brain, which is interesting. And then we have a coronal slice here. And we have one of the hallmarks of Alzheimer's, which is the hippocampus atrophying. And this helps explain the poor word recall we saw earlier, as it's responsible for the consolidation of short-term and long-term memory. You can see that here. Okay, so we have some basic questions for everyone, whoever just wants to chime in. Why is the ventricular system affected even though it's a non-neuronal structure? These are questions from the text.
So it's not that it's necessarily affected, it's the cortices that are affected, they're atrophying, so then the ventricular system expands.
Exactly, yeah, and this is called hydrocephalus ex vacuo, to put a term to it. So the skull volume is fixed, so any decrease in neural tissue means an increase in ventricular volume. Okay, are some brain areas affected more severely affected than others, and which ones are they? You want to quickly answer that? I guess maybe people's mics aren't working so well. Is the chat on? Oh, there is a chat. Okay. I think I'll still stay in full screen so that people can see. But it means I can't see the chat, so you'll have to speak or someone will have to tell me what the chat is saying. Okay. In any case, that was a general overview case just to show how a global change to the brain affects it. There's obviously so much more to get into, but this chapter covers basically the groundwork for everything we'll be diving into later. And to understand the brain, the chapter lies out that we have to first understand where the structures are, so spatially where they are, and this is called regional neuroanatomy. And then we have to understand how they work together, and that's called functional anatomy, or what the structures do. And the combination of this is functional localization, which is really the goal of Martin and of this whole lecture series. So if we can get through all of that, it'll really help us as future clinicians or current clinicians to localize and treat neurological disease. We'll start with the basic cellular components and then go into the major regions and functions that Calvin especially will be taking over. Okay, so first we have the fundamental concept of the neuron as the functional cellular unit of the nervous system. This was actually discovered in the late 1800s with Ramani Cajal, which we'll be going back to later when we talk about the different types of cells. And 100 billion neurons are in an adult brain, which connect together to perform myriad tasks. Here is a general representation of a neuron, and the different parts include the dendrites, which receive info from other neurons. In particular, in the dendrites, there are spines, which are actually contacts for excitatory synapses, and they're extremely plastic and are implicated in memory and learning. Then we have the cell body, which is the metabolic center. This involves the nucleus and organelles, and it actually receives input from other neurons and integrates information as well. Then we have the axon, which conducts action potentials. And this leads to the axon terminal, which connects two neurons together at the synaptic cleft. And synapses can actually be formed on not just the dendrite, but also on the shaft of the axon, the cell body, and the initial segment. Just a quick question for everyone. Does anyone know where the Nissl stain shows on the neuron, or what particular parts of the neuron it stains? Well, the answer would be the rough endoplasmic. Oh, sorry, is someone saying something? It would be the rough endoplasmic reticulum. I don't think I can hear anyone's input, but these are the overall structures, and they're actually different types of neuron. There's so many different types, but we're going to put them into three main classes based on the numbers of dendrites and axons. So the unipolar, the bipolar, and the multipolar. First, we have the unipolar neuron, which is the simplest with just a single axon giving rise to multiple processes. These are the least common, but control exocrine gland secretions and smooth muscle contractility. Then we have bipolar neurons, which have two processes coming out from different sides of the body. And these are many of our sensory neurons like olfaction and touch. And this is actually a representation of the bipolar cell of the retina. Included in that is a subset called pseudo-unipolar neurons, which include a subset of two processes that fuse into a single process early in development. And this is actually the neurons we see in the spinal cord, the ganglion cell of the dorsal root. And it's involved in transmitting touch, pressure, and pain. I took these images actually from a different textbook called Candel. It's used widely in neuroscience. Lastly, our third class would be multipolar neurons, which are actually a complex array of dendrites, or they have a lot of dendrites and a single axon that branches extensively. And these are the majority of the neurons in the brain and spinal cord. This is a motor neuron in particular, and they can be projection neurons. This is a subclass, which means that the axons reach to far off places up to, say, 1 meter long. This is how the central nervous system connects to peripheral targets. I just also want to show that it comes in many different forms. So this is the pyramidal cell from a hippocampus. And we also have a Purkinje cell from the cerebellum, which has up to 1 million dendrites. So multipolar cells, as well as every other type, come in many different shapes and forms. Okay, now getting to the synapse in particular. This is where information from one part of the neuron transmits to another. And there are 3 main parts. The presynaptic terminal, which is the axon terminal of the first neuron, the synaptic cleft, the space in between, and the receptive membrane, which hosts the dendrites of the postsynaptic neuron, as an example. So the way broadly this works, and of course we could go into greater detail, is that an action potential triggers a voltage-gated calcium channel in the presynaptic neuron to open, and the influx of calcium causes vesicles to fuse and release neurotransmitters. These diffuse across the cell, the cleft, and bind the postsynaptic membrane, which ultimately changes the permeability of ions at the receptive membrane. And many chemicals act as neurotransmitters with different effects. For example, glutamate and acetylcholine are excitatory, and GABA and glycine are inhibitory. Now, if a neuron is inhibitory, what ion does it cause to change in the postsynaptic membrane? Can someone answer that?
Chloride.
Yeah, so chloride would help depolarize, and you would depolarize with sodium. So if there is a direct change in ion influx, that's called an ionotropic receptor. So there are different types of receptors. Different neurotransmitters like dopamine and serotonin can have different actions, even on the same neuron, just based on what receptor subtype there are. So the other main subtype of receptors are called metabotropic receptors, which involve secondary messengers, often with G proteins, and a bunch of complicated downstream effects, which can either just change the ion permeability as well or cause different types of gene expression. So dopamine subtype 1 receptor is actually depolarizing, while the second type is hyperpolarizing. Okay, and here we've been describing neurotransmitters as chemicals, but actually there are also just synapses that are electrical, which means that a neuron just connects to another by a gap junction, and there's no chemical intermediate, which is kind of interesting as well. Okay, so we talked about neuro, but glia actually outnumber neurons 10-1. So they're hugely important and are responsible for our structural and metabolic support. There are 2 major classes, microglia and macroglia. So first, microglia. They are our phagocytic scavengers. They respond to infection and damage. These are what they look like, and here's a single one. They can destroy microorganisms, clean debris, repair tissue. They release inflammatory mediators, and unlike the rest of the glia, they're from the bone marrow. So they're mesodermal and are really the macrophages of the nervous system. Next, we have macroglia, which includes the oligodendrocytes, Schwann cells, astrocytes, and ependymal cells. So oligodendrocytes and Schwann cells form myelin around nerves, which increases the action potential velocity and its conductance. And that's what gives it its white look. So oligodendrocytes wrap around nerves in the central nervous system, and one oligodendrocyte can wrap multiple nerves. Can someone tell me, are cranial nerves wrapped by oligodendrocytes?
So I could be wrong, but I think in general they are wrapped by Schwann cells, but I think cranial nerve 2 is wrapped by oligodendrocytes.
Perfect, yeah, so cranial nerve 2 is actually wrapped by oligodendrocytes, whereas 3-12 are wrapped by Schwann cells and 1 isn't. Very good. Okay, great. So this is the predominant glia in white matter, and some pathologies that you can think of with oligodendrocytes include multiple sclerosis, PML, leukodystrophies, just to name a few. So here's just another look of an oligodendrocyte in vitro. Now, Schwann cells wrap around peripheral nerves, and this includes cranial nerves 3-12, as I think Jake said. And they only wrap around one nerve, and they're actually from neural crest origin. And a pathology we can think of is Guillain-Barré. All right. Next, we have astrocytes. Astrocytes, it's the one in green, and they have so many different functions. They can act as scaffolds for growing axons. They're fuel reserves for glycogen. They connect to synapses and regulate glutamate and potassium. There are just so many that they do, but a really important function is in the blood-brain barrier, where they form, where the foot processes help contribute to the blood-brain barrier. Next, we have ependymal cells. These line fluid-filled cavities in the ventricles in the CNS. So this is a ventricle here, and you can see the ependymal cells lining it. And here are some of the individual cells. They are ciliated, simple columnar, and the cilia circulate the CSF while the microvilli help with absorption. One special type of ependymal cell is in the choroid plexus that produces CSF, just as an interesting correlate. All right, so now we're building towards the general organization. I want to talk about the development of the brain and spinal cord. So we have gastrulation, which ends in the trilaminar disc. The notochord, which is actually from mesoderm, induces the overlying ectoderm to form neuroectoderm and neural plate. This forms into a tube-like structure called the neural tube as the neurons and glia proliferate, and the walls form the structure of the CNS while the cavity forms the ventricular system. This is taking it back to really basic science. The neural crest from the top of the roof migrates out and forms many other cells in our body. And this organization is actually really important because the ventral aspect becomes the basal plate, which actually is involved in motor functions, and the dorsal aspect is the alar plate, which is involved in sensory functions. And the brain, the whole CNS actually retains this organization. And just as an example, other than the spine, you can see in this brainstem too that this organ is largely retained. Okay, after the neural tube forms, this rostral portion forms initially 3 hollow swellings called vesicles. There's the forebrain, the midbrain, the hindbrain, and the spinal cord, which is largely undifferentiated. From a lateral view, you can actually see flexures as well. And these are from the growing cells, which gives the maturing brain its complex configuration. There's the cephalic flexure, which is at the level of the midbrain, and the caudal flexure, and the cervical flexure, which is at the junction of the spinal cord and the caudal hindbrain. At the 5-vesicle stage, the forebrain becomes the cerebral hemispheres and the thalamus and hypothalamus. The midbrain develops, the hindbrain becomes the pons and medulla, and you have the spinal cord. And now we also have an additional pontine flexure that becomes prominent. But by birth, all of these flexures straighten out, and it's just the cephalic flexure that remains and forms the bulk of the brain. Okay. Now, overall organization. The nervous system is divided into 2 parts, the peripheral and the central. So we'll start with the peripheral. It's what's outside of the brainstem and the brain and spinal cord. And it is further divided into the somatic and autonomic. So the somatic division involves sensory neurons, such as the ones innervating skin, muscles, and joints, as well as motor neurons that innervate muscle. And then we have the autonomic system, which innervates glands and smooth muscles of the viscera and blood vessels. And this is further just subdivided into sympathetic, parasympathetic, and enteric subdivisions. Now in the peripheral nervous system, the cell bodies collect in the peripheral ganglia, and the axons are in the peripheral nerves. There's so much more to go into, but that will be covered in the later chapters. Now the central nervous system is the brain and spinal cord. And the brain has 6 regions: the medulla, pons, cerebellum, midbrain, diencephalon, and cerebral hemispheres. And then including the spinal cord, that's 7 divisions. In the central nervous system, the neural cell bodies are located in lamina, these flattened cortical sheets of cells, and the deeper clusters beneath them can form many shapes, and they also are nuclei as well. Okay, now to the spinal cord. Some of the main functions involve processing sensory information, controlling body movements, regulating visceral functions, and transmitting both ascending sensory and descending motor. So it's the only part of the CNS with external segmental organization, and it has a basic structure. So there's a pair of nerve roots in the dorsal and ventral roots. They anatomically separate the sensory motor components. So the dorsal roots contain only sensory axons that send signals into the spinal cord. Does anyone remember what type of neuron this is out of the basic 3 types of neurons that we covered earlier? So like unipolar, bipolar, pseudo-unipolar, multipolar. Okay, the answer is pseudo-unipolar, and it's actually also from the alar plate. The ventral roots contain motor axons that sends out commands from out of the spinal cord. There's an exception though. Cranial nerve 11 is the only motor root to rise dorsally. It's also special because it's uncrossed. Okay. Quickly moving on. The brainstem is composed of 3 divisions, the medulla, pons, and midbrain, and it has 3 general functions, which involves receiving sensory information. It's a conduit for information that flows through it, such as the ascending and descending tracts, and it integrates information. And that's what's different about the brainstem versus the rest of the spinal cord. It has higher order processing that is unique to it. And we'll also talk about the cranial nerves in depth much later, so I won't go into it in depth now. Additionally, each of the subdivisions here serve many different functions, and just some of the functions to say, medulla covers brain blood pressure, respiratory regulation, and damage to it's almost always life-threatening. The pons and midbrain are partially involved in eye movement and so much more. Brain, pons, and medulla. Lastly, we have the cerebellum for my part, and it covers 5 general functions: limb movements, eye movements, it helps maintain posture, participates in higher brain functions, and in particular, if you damage the cerebellum, particularly the lateral hemispheres, they cause limb movements to become poorly coordinated. Okay, so that's my half of the presentation. I'll now hand it on to Calvin. I'll just stop sharing my screen so you can take over.
Okay. Yes, cool. Nice. Okay, I'm Calvin. I'm a first-year med student. I went to Williams. I'm coming to you from Poughkeepsie, New York. We're quarantining over here. Things are good. So one thing I want to mention is that I have like 17 screens set up right now. So I can see the chats on my iPad. So we should have planned that ahead of time, but that's okay. All right. So what I'm going to try to do is continue on this track of looking at both the anatomy and some of the basic functions of what's going on in the brain. So to start off here, we have the diencephalon, which is composed of both the thalamus and the hypothalamus being below the thalamus. And really what it does is it serves 3 basic functions, which are sensory functions, motor functions, and integrative functions. But really we can appreciate the breadth of functions that even just those 3 things compose: sensory, motor, and integrative. And it's these integrative functions that make the diencephalon unique from the lower divisions that Andy mentioned earlier, the midbrain, pons, and medulla. So here I'll start off at the thalamus. It's a key structure that is really important in transmitting information from this to the cerebral hemispheres from the body and the lower brain structures to sort of have that pathway up to the upper parts of the brain. And it's composed into sort of sections called nuclei that are associated with different cortical areas. So it's kind of compartmentalizing the information that's being delivered upwards. The thalamic adhesion is a small portion of the thalamus at each half between the 2 hemispheric halves of the thalamus that you can see in this image over here. And then I want to mention that the thalamus is participates in the endocrine system, and it releases hormones from, or stimulates hormone release from the pituitary gland. Here I wanted to give you a sense of the nuclei of the thalamus and to give you an idea of the diversity of transmission functions of the thalamus. I'm not going to go through this, but just looking very basically, we see pain, pressure, face sensation, vision, hearing, motor. So really the thalamus is a very important structure in integrating information and passing it up to the upper parts of the brain. So two hemispheres, two cerebral hemispheres that compose the brain. These are the most highly developed portion of the human CNS. So 4 major components here that I've boxed and also listed here: the cerebral cortex, the hippocampus here, the amygdala, and the basal ganglia. And I've starred those that are considered subcortical, and we'll focus on those first. So the amygdala here is really important for emotions and the response to stressful or threatening situations. So it'll be activated during those periods of times. And the hippocampus, I use this nice mnemonic here. I don't know if I made it up, but need to do a lot of memorizing on campus, hippocampus. So, you know, learning and memory with the hippocampus. And then the fornix and the mammillary bodies are anatomically and functionally related to the hippocampus. You can sort of view it as this extension of the hippocampus. Now, a brief aside, the hippocampus and the amygdala are part of what's called the limbic system, and it includes these other structures as well down here, which I won't name, but I really like this mnemonic here, the 5 Fs: feeding, fleeing, fighting, feeling, and sex, which you can fill in the last F for that one. So that's the limbic system, and the hippocampus and the amygdala participate in that. Moving on to the striatum, which is part of this basal ganglia. It's important in movement. The basal ganglia is important in movement, cognition, and emotions, which I have here. The striatum participates in that as well, has this really interesting complex shape that I hadn't seen this brain structure before making this presentation, so I'm interested to learn more about it later. The ventricular system was briefly mentioned earlier with the clinical vignette in the beginning. It's a place where the CSF accumulates. It extends into the spinal cord as the central canal. The CSF exits here and bathes the entire surface of the CNS. I'm going to touch on this a little bit more later. A quick clinical correlate with the basal ganglia. As I mentioned, it's really important. The basal ganglia is really important in coordinating movement. So I just have these pathways here. There's direct and indirect, inhibiting and facilitating movement. So in the basal ganglia, if you lose some of these dopaminergic neurons, which manifests as depigmentation of the substantia nigra pars compacta, which is part of the basal ganglia, we have severe motor deficits, and Parkinson's at its in its essence is a motor disorder. And a histological finding, a pathological finding here are these Lewy bodies composed of a protein called alpha-synuclein. Another clinical correlate briefly I want to mention is holoproencephaly. It's an embryological defect where the embryonic forebrain fails to separate into these 2 cerebral hemispheres. And I noticed that here the basal ganglia, here the star is fused, and that's visible on MRI. Now some physical features of the brain here, the gyri and sulci. Not sure if I'm pronouncing that right, but the gyri are the bumps, and the sulci are the grooves. And those are very apparent. Here are the gyri, and here are the sulci. Okay, the brain has some of these important fissures and sulci that I want to point out here. First is the lateral sulcus, dividing the brain here in between the frontal lobe and the temporal lobe here. The central sulcus here divides the parietal and the frontal lobes. And then the sagittal fissure, I think fissure is really telling you that it's a bit deeper or more important than some of the other structures. And it's this sagittal fissure that separates the two cerebral hemispheres. Okay, now the cerebral cortex, which is basically this upper helmet part of the brain, has 4 lobes. You may have heard this before, but the frontal lobe, basically at its essence, is thought, action, cognition, and emotion. Parietal, touch, pain, proprioception, which is the awareness of where your various body parts are and how much they're stretching or not. The temporal lobe, speech, hearing, memory, and emotion. Then occipital is a bit unique in that it only really is involved in vision. It's kind of selfish in that way that it only controls vision. But we as humans have very good vision, so we need a big occipital lobe devoted to it. Okay. Now, here I'm just pointing out different views of these 4 lobes: the frontal, temporal, parietal, and occipital. And this is the inferior view looking up from the feet, and here from the top. So I just wanted to give you a sense of where these are. Now, facts about the frontal lobe. First, we'll start with the primary motor cortex, really important part of the frontal lobe. It controls the speed and directions of mechanical actions of movement. It's located in the precentral gyrus of the frontal lobe. The premotor areas, as an aside, which control decision-making and planning of movements, are located in the superior, middle, and inferior frontal gyri, adjacent to the primary motor cortex. And then the inferior frontal gyrus is where Broca's area is located. I have a picture of that later on, which is important in the articulation of speech. Now moving along to the association cortex. Much of the frontal lobe is association cortex. You can think of this as the biggest part of the association cortex. It's really important in complex processing and integration of information from different systems and modalities. And the prefrontal association cortex is important in thought, cognition, and emotions. The cingulate gyrus here, and the medial frontal lobe and the orbital gyri, which I've pointed out here, this groove here, and then the cingulate gyrus is here. This sort of surrounds the ventricle there and the corpus callosum. These are really important structures in emotion. The basal forebrain uses acetylcholine to regulate cortical excitability. And finally, the olfactory bulb right here is important in smelling things. Important to note is that the olfactory bulb and tract project into the temporal lobe from the frontal lobe, even though they're directly beneath the frontal lobes. And these neurons are also the only ones that sense and do not synapse in the thalamus. So they sort of bypass the thalamus. Okay, now the parietal lobe, facts about the parietal lobe. The primary somatosensory cortex, or the primary somatic sensory cortex, located here in the post-central gyrus as opposed to the pre-central gyrus, is the initial cortical processing area for sensory input. So the parietal lobe, like I mentioned, is really important in sensation and touch. So it's here in the primary somatic sensory cortex that some of that stuff begins. The remaining parietal lobe on the lateral surface of the hemisphere is divided by the intraparietal sulcus. I'm not sure that's on, oh, it is right here. Into the superior and inferior parietal lobes. The superior parietal lobe up here is involved in higher order somatic sensory areas and other sensory areas. And then finally down here, the inferior parietal lobe lobule or is involved in integrating diverse sensory information to allow for processes such as perception, language, mathematical reasoning, and visual spatial cognition. And a quick clinical correlate, lesion of that inferior parietal lobule here results in a syndrome called Gerstmann syndrome. And it's these features agraphia, the inability to write, acalculia, difficulty in learning or comprehending mathematics, finger agnosia, the inability to distinguish fingers on the hand, and then left-right confusion. So this structure here is of importance in that way. Now, the occipital lobe, which I mentioned, is sort of selfish in that it only is involved in vision. It's separated from the parietal lobe on the medial surface by the parieto-occipital sulcus right here. There are no distinct boundaries on the inferior and lateral surface, just an imaginary line between the preoccipital notch and the parieto-occipital sulcus, which I think we'll see in another picture later. The primary visual cortex, which is the initial cortical processing area for visual input, is located in the walls and the depths of the calcarine fissure on the medial, right here, the calcarine fissure of the medial occipital lobe. So that's where the primary visual cortex is located. The surrounding higher order visual areas allow us to perceive color and form from raw visual input from the eyes. Now, the fusiform gyrus, I don't have that on this picture. Also known as the occipitotemporal gyrus, is located on the ventral brain surface and is really important for recognizing faces. So patients with lesions here can confuse faces with inanimate objects, a condition called prosopagnosia. Facts about the temporal lobe now. The temporal lobe, like I mentioned, it mediates hearing, some components of vision, memory, and emotions. And it's separated from the frontal and parietal lobes here by the sylvian fissure. So now the primary auditory cortex right here, it's located on the superior temporal gyrus and allows for the perception and localization of sound. The the posterior part of this superior temporal gyrus, right here, of the dominant hemisphere, which if you're a righty is your left side, is specialized for speech and is known as Wernicke's area, which I will show you later. And a lesion of this area impairs the understanding of speech, whereas Broca's area before was the articulation, the generation of speech. Okay, the posterior middle temporal, here we go. The posterior middle or medial temporal gyrus is important for the perception of visual motion, and the inferior temporal gyrus is important for the perception of visual form and color. The temporal pole also important in emotions. Okay, temporal lobe. Now the insula. I mentioned the sylvian, fissure. This insula is sort of buried underneath, and it's located within this fissure. It's covered here by the frontal, parietal, and temporal lobes. It's buried by the opercula of the other lobes in the 8th month of prenatal development. I have a slide later on that sort of shows the insula being covered up. Its functions are in taste, internal senses, pain, and balance. Okay, here's that other picture that I mentioned a gazillion times. I like this one because it shows you Broca's area here in the frontal lobe, and then also the primary auditory cortex here, the primary visual cortex in the occipital lobe, Wernicke's area, which is important for understanding and comprehension of written and spoken language, Then here, that somatosensory association, the premotor cortex. Like I said, I like this picture a lot. Okay. Now, the corpus callosum, and in general, structures or tracts of containing axons that interconnect the two halves of the brain are called commissures. Now, the corpus callosum is the largest or most important of these commissures, and it connects the two hemispheres of the brain. And I do want to mention the 4 parts of the corpus callosum here: the body, the splenium, the genu, and the rostrum. Information from each lobe, as you might imagine, travels through the adjacent segment of the corpus callosum to the contralateral or other-sided lobe. For example, information from the occipital lobes travels through the splenium, which is this rear or rostral part of the corpus callosum. Okay. Ventricular system here, we mentioned before, it's made up of fluid, filled cavities or ventricles, so they're basically empty spaces, connected via channels. Both are within the brain and contain CSF, like we mentioned. Andy talked about the choroid plexus already. It's responsible for the secretion of CSF. The lateral ventricles here are located within each cerebral hemisphere. The third one, the third ventricle, is located between the two halves of the diencephalon, the hypothalamus and thalamus. And then the fourth, down here, is between the brainstem and the cerebellum. Okay, and one other thing, the CSF is really important because it cushions the brain and allows for chemical communication. Here's just another image of the ventricular system, and I like this one because it shows the boundaries between each of the ventricles. The intraventricle foramen of Monroe, which is here, interconnects the lateral and the third ventricle. The aqueduct of Sylvius here connects the third and the fourth ventricles. And then the ventricular system extends into the spinal cord as the central canal. Clinical correlate quickly, bleeding into the ventricles is called a neonatal intraventricular hemorrhage. And these babies have increased risk. Babies who are premature and low birth weight have a greater risk of this. And these infants present with altered level of consciousness, bulging fontanelle, hypotension, seizures, and comas. It's a pretty serious condition when these ventricles are hemorrhaged. Okay, the central nervous system is covered here by these 3 meningeal layers that you may have heard before, and I have this nice in to out flow here. So the pia first is a really thin, fibrous inner layer that covers the CNS. It firmly adheres to the brain and the spinal cord. It's derived from the neural crest. The arachnoid mater is the middle layer. The reason it's arachnoid, which might make you think of spiders, it's because it contains these web-like connections to the pia that literally look like a web. And the dura mater, which is a thick outer layer closest to the skull. And unlike the pia and the arachnoid, it's derived from mesoderm. The potential space between the arachnoid and the dura is known as the subdural space, underneath, subdural. This is where subdural hematomas collect when dural veins are torn after trauma because the arachnoid is exposed but not adherent to the dura. I mentioned that pia is delicate, adheres to the brain. The space between the pia and the arachnoid is important. It's known as the subarachnoid space. It's the location of the cerebral spinal fluid. The thin filaments of the arachnoid give it its name because they're reminiscent of a spider's web, like I said. Okay, extensions of the dura are important. The first is this falx cerebri. It separates the 2 cerebral hemispheres, sort of right down in the middle. The tentorium cerebelli is a smaller structure down here, separating the cerebellum from the cerebral hemispheres. And these are both extensions of the dura. Another clinical correlate, I'm not going to go into this in great detail, but just to mention that depending on where, in between which of the three meningeal layers the hemorrhage happens, has a different appearance on imaging. So this might be covered in later lectures, but each of these four have distinctive imaging characteristics that clue you in on where, in relation to these three meningeal layers, the hemorrhage or hematoma is occurring. Now, human brain development wasn't covered too much in this chapter. The important bits that I picked up on were first this emerging C shape of the ventricle. I kind of think of the brain as starting out straight and then it sort of curls into this C shape. And then finally, I alluded to this before, but the insular cortex initially on the surface is buried at about the 8th month of development. Okay, some terms that are important in describing our anatomy here. First, to note is the central nervous system is organized along two axes of the body, which are depicted here in this nice mouse. The rostral caudal axis. I did misspeak earlier, by the way. It's caudal part of the corpus callosum. Okay, anyway, the rostral caudal part of the central nervous system, and then the dorsoventral axis of the central nervous system. And here in the brain, it's sort of just twisted along this axis. So rostral up here towards the head, and then caudal down towards the feet, dorsal towards the back, ventral towards the stomach. Okay, anatomical planes here, horizontal or axial planes, sort of as the name implies, horizontal. The coronal plane here is oriented like this, and then the sagittal plane sort of cutting them right in half. And then one other point is that the sagittal slices that are exactly in the midline are called mid-sagittal. And then whereas if they're not in the middle, they're called parasagittal. Okay, that concludes the content portion of the presentation. We did include a bunch of questions that were included in the end of the text. I see the chat, so if you guys, you know, type in, there'll probably be a gazillion responses, but that's okay. I'll see what the consensus is and give you a couple of seconds to
I guess before we go into that, does anyone have any questions on the presentation earlier for either of our parts?
Yeah.
Or things they wanted to add, elaborations, anything like that? Give it a couple seconds.
No, I'd say great job putting these talks together. I think you corrected yourself, Calvin, that the splenium is the most caudal part of the corpus callosum. That's really it in terms, I think, you know, most of it was pretty spot on. We do have a little bit of time, so if you want to do some questions, I think that would be great, and then we'll just kind of end at 3 o'clock. Obviously, the questions are good for people to kind of do on their own as well.
Oh, that's great. Okay.
I guess, and I'll just, for anybody that wasn't available when we first came on, I just wanted to say that this is kind of a model, I think, for how you guys can put your presentations together. If you, I send out the email to download this video, you can watch this video and read through the first chapter. And what Andy and Calvin did is they really followed the chapter exactly as it is written. They integrated parts in different areas of the text that are related to sort of create, kind of talk about things in place, and so that it has a good linear flow, and then they integrated the figures really well. So I thought it was a really good talk. And what we will do is you guys can send me the slides 2 weeks before you do your presentations, and I can kind of review them, as can your resident mentor. I'm doing that basically so that if you don't have a resident mentor, we can make sure that you get some resident feedback. I talked to Jade, the PGY4, PGY4, and so she's going to sign up for some projects, some chapters over the summer that will take place over the summer. So there are more people hopefully that will be signing up. But that way, if you at least send them to me 2 weeks before you give your presentation, I'll be able to help you guys out if you don't have somebody assigned to you. And that gives me enough time to do that. And then the second part of this is hopefully people come to these having read the chapter. You get to see the lecture as kind of a second way, an audiovisual way to reinforce what you read. And then we'll be sending out these Anki slide decks, and you guys will be creating those for each of your chapters. And I'm creating the one for this section. And you'll be able to see kind of how I put that together. And then hopefully you can use those to integrate the information over time and kind of keep it in your memory. And hopefully review everything that we've gone over time, because that's kind of the way the program is designed to help you learn and retain information. So why don't we do some questions? And if there's any questions, you guys can obviously always email me about, the flow of things. Next week, I already said Dr. Wu will be doing a gamma knife talk. So the people doing chapter 2 will have still have 2 weeks if you send me your PowerPoints, sometime this week. Okay, so why don't you guys take it away and go through some questions? I don't know how you've divided them up. If Andy, maybe you want to do one, and then Calvin will do one.
Sure, yeah. I can just do the ones that relate to me, and then Calvin can do the one.
That's perfect.
Calvin, do you want to just maybe use your screen if that's okay?
Yeah.
Just to save the hassle. Sweet. So which of the following analogies best describes the functional relationship between two different parts of a neuron? I'll let you read the rest. Maybe give like 15 seconds for people to type in their answers. Okay. Maybe do you want to reveal the answer? All right, so a dendrite is to an axon terminal as a presynaptic input. Signaling is to a postsynaptic output. So really just labeling the right parts of each component. So here's the synapse with the axon terminal and the dendrite. The axon terminal being the presynaptic, dendrite being the postsynaptic. Great. Which of the following neuron components is located within a white matter tract? The dendrite, the cell body, the axon, or the axon termination? And just give like another 10 seconds. Yeah, okay, so I see some answers. C, the axon, perfect. So, which of the following best, okay. Which of the following best lists the parts of the neurons that are located in the nucleus? Couple more seconds. All right, yep, so let's reveal the answers. The app. Wait, Calvin, did you get?
The nucleus are these, this might be actually my portion, because the nucleus, I think it's referring to the thalamic nuclei, and there are more like sections of this entire section of the brain. So it indeed does include all 4 of these structures.
Interesting. I guess
not nucleus of the cell, nucleus
right.
of the thalamus.
Interesting. So this question is maybe a little, I think a lot of nuclei actually only include the cell bodies, and then you have, at least, maybe that's a different type of nuclei. maybe not what they're trying to go for. Okay, next question. Which of the following best describes the function of Schwann cells and oligodendrocytes, respectively? Okay, should we just reveal the answer? Nice. So the myelin sheath of peripheral axons and the myelin sheath of central nervous axons. So the image from earlier, oligodendrites in the CNS and cranial nerve 2, and Schwann cells in the peripheral nervous system, including cranial nerves 3 to 12. The person suffers from a traumatic brain injury. There is bleeding and inflammation at the injury site. Which of the following cell types plays a phagocytic role in eliminating blood and tissue debris? Nice. So Jake and Alpha are right, the microglia. They are the scavenger cells, the macrophages of the nervous system. Ependymal cells are located in which of the following nervous system structures? Yep, so the ventricles. And Calvin included a nice picture of the...
Yeah, sometimes they slough off when doing a lumbar puncture, and if you have an infection or a subarachnoid hemorrhage. So these kind of, they actually, the source was saying they kind of look like malignant cells, but they're not. They're just ependymal cells. Okay.
Okay, which of the following is not part of the peripheral nervous system? Nice, okay, so yes, the motor neuron cell body. Here's just the spinal cord.
Yeah, I just wanted to point out where this anterior ventral horn is coming from, and it's white.
Gray, actually.
Gray, yeah, actually it's one of the gray matter.
Cell bodies are, I mean, it looks lighter on this stain, because I think I don't know what the stain is, but I think they're staining probably for myelin here.
Right, the outside are the white matter with the tracks,
right.
and the cell bodies are on the horns.
Gray.
The
white.
gray matter, yep.
Okay.
Which of the following is not a feature of the autonomic nervous system?
Yeah, this is a...
Yep, innervation of somatic motor neurons that innervate limb muscles. That would be... okay, which of the following best describes the cell? Oh, all right, this is your part.
Yeah, this is my part. Which of the following best describes the sulci and the gyri? Yep, see the gyri are the bumps and the sulcus here is the groove. Okay, which of the following best describes the location of the major brain regions? These are anatomical relationships here. Yeah, that's right, A, thalamus. The thalamus is above or rostral to the midbrain. Okay. A patient has a tumor in the region of the insular cortex. Which of the following best describes the location of the tumor? Yep, that's right. It's buried beneath the frontal parietal and temporal lobes. It's sort of tucked in there. A pitcher was hit in the head with a baseball. Not right now during COVID. The impact of the ball hitting his head caused a skull fracture over his left orbit. Which of the following brain structures is located closest to the site of fracture? I'll give the, yep, that's right, the inferior frontal lobe, bam, that's where it happens, just above the eyes. The falx cerebrae separates the That's right, the two cerebral hemispheres right here, this extension of the dura, the tentorium cerebelli being the other one, separating the cerebellum from the hemispheres. Last two, the atrium of the lateral ventricle is located within which major CNS division? This is the cerebral cortex. This was the lateral, yeah. So it's here in the cerebral cortex. Okay, finally, a brain MRI in the coronal plane would not, Calvin. Would not image on a single slide which part? Well, I'll give it to you. It's the frontal lobe and the occipital lobe here on the coronal plane, because the frontal lobe is up here and the occipital lobe is in the back, so you can't get it all in one. Okay, right on the dot. Thanks, guys.
Thank you,
Great.
guys.
Thanks, everyone.
We'll see everybody else next week at 3 o'clock, sorry, with Dr. Wu. And we'll be talking about Gamma Knife. And then the following week, we'll do chapter 2. Okay? Thanks, everybody.